we need to discuss a little bit about our relationship to disease-causing organisms. And so we're going to discuss like the principles, what makes something pathogenic, why do we regard things as being pathogenic, and what is their relationship to us as human beings, remembering, of course, that all living things from bacteria on up are subject to infection by parasites, by pathogens, which take them over and subvert all of their functioning and effectively cause disease. Even bacteria, for example, have viruses that infect them. So we're going to, first of all, we need to get some definitions down. In this class today, you're going to hear a lot of these definitions. I know it's not the most interesting thing on earth, but it is really important because it allows us to conduct the conversation in epidemiology and everything with much more facility. So the pathology of a disease is the study of the disease itself, of what it does to an organism. Etiology of disease is the derivation of the disease, the cause. Not only the organism, for example, which causes the disease, but why? Why did it enter a particular host population? How did it enter? The pathogenesis of a disease is the study of the development of the disease in the organism. It's the course of disease, the course of the illness, if you like. This term infection we use very loosely, but I say this because when we say that it's simply colonization of a body or invasion of a body by something, that something is not necessarily causing disease. And this word infection applies in particular to things which cause illness. In other words, things which are pathogens. And we're going to discover in a little while that what makes these organisms pathogenic is that they are parasitic. That is, they in invading a body or invading an organism, what they are doing is they are using that organism entirely to their own benefit. And they are affording no benefit to the host. You'll see exactly what I mean in a few minutes. But they afford no benefits to the host. They only cause damage. And that is the definition, essentially, of both a parasite and a pathogen. All pathogens are parasites. A disease, the word disease, it seems so familiar to us. But in actual fact, it has a very specific meaning in microbiology and in science. It means an abnormal state. It is a departure from homeostasis due to some specific cause. So a disease, abnormal state where the body is no longer performing its normal, proper functions. So here's what I meant when I said that we need to talk about our relationship to these microorganisms and other things around us. We, in fact, have a very intimate relationship with a large number of microorganisms which never cause us disease. And we need to remember that disease-causing organisms are by far the vast minority of microorganisms compared to the huge number of microorganisms that are out there in the environment that cause no disease process whatsoever. The number of microorganisms which cause disease, especially in humans, is very, very small. It's a very small percentage of the total number of microorganisms that there are. And the first thing to realize is that we ourselves have a population of microorganisms which inhabit us. They inhabit almost all of us, outside and inside. The outside, our skin, all of it, the outside of us is really quite densely populated with microorganisms. And these are a perfectly normal part of our makeup. In fact, when we disturb them, we often get ourselves into trouble because they are a protective mechanism. This is our normal microbiota. That microbiota, by the way, may be quite changeable. We may have things which are permanent, which we could always find on our skin, for example. And then just temporarily, we may have transient populations of things coming in and out. They may cause some change that we might even notice, but they cause no damage. An example, for example, we may have fungi that temporarily occupy space. You may notice, for example, changes in your body makeup. Maybe your breath changes because you've got a change in the microbiota in your mouth. Maybe your body's odor changes because you have a temporary change in the microbiota that cover your skin. So first of all, we have a healthy, living microbiota on the outside of us. The next thing that we need to think about is our interior, what we mean by our interior. First of all, you need to realize we are like a tube. We're like a thick-walled tube with a hollow running right through us from mouth to anus. That is our gut, comprises our gut cavity and also our respiratory cavity, and then reproductive tract as well. So we have our outside, and then we have this inside. I would like to call it the inside outside because it communicates with the outside. But then inside the body itself, we have an interior part where all of our organs are. And that part is sacrosanct. That part should not have resident bacteria or anything else. And as far as possible, it is kept sterile. But our gut cavity, much of our respiratory tract, in women's reproductive tract, these are all populated by a rich population of normal human microbiota. As I say, it's not a fixed entity. It changes. It changes with all sorts of reasons. It is a dynamic thing, but it is terribly important because it is protective of us, as we'll discuss in a second. So these are just some illustrations from your book of various bacteria, part of the normal microbiota. Here, for example, in the nose, very, very rich in bacteria. These are cocci, and probably they look like streptococci, which are very, very common in nasal passages. These are spiral sort of bacteria from the lining of the stomach. A very, very harsh environment because it's an acid environment. There's not a lot of bacteria that survive in the stomach, actually. And there's not a lot of bacteria that we swallow that survive passage through the stomach. Nonetheless, it has its own resident flora. And then in the intestines, the intestines have a huge population of bacteria, a very, very diverse, very active population of bacteria, which do all sorts of things for us. They are by no means just hanging out and making use of our food and everything else. In fact, we cooperate with these bacteria intimately, and we're discovering that there are many, many ways, strange ways, in which the gut flora especially contribute to our well-being. Changes in gut flora are being traced, for example, to changes in mental status, extraordinary as it might seem. They contribute vitamins, they participate in nutrient dynamics, all sorts of things. They're an essential part of our existence. Now that all of this rich microbiota, we used to think that it only began to be established once an infant was born. It now turns out that in fact, the microbiota begins to be established in utero, that the uterus is not, and the fetus, the infant itself, are not actually sterile, as sterile as we always thought. It is not absolutely clear how bacteria make their way to the fetus, but in fact, it appears that they do. Gut content of the fetus at birth, for example, is not entirely sterile. It doesn't have a very rich microbiota, but is not sterile. An infant begins to establish its microbiota during birth. Its first exposure to major populations of bacteria is by vaginal birth. And as we'll see in a second, there are groups of bacteria which are specifically associated with that process. Once the infant is born, the microorganisms that the child acquires, it acquires from its environment, from the people around it, especially if it's being breastfed, for example. It rapidly builds up a very rich gut microflora, and that remains with us throughout our life. Dynamic, changing, but nonetheless normally always present. So let me just, sorry. Here, you absolutely don't need to remember all of these. These tables are simply to show you the incredible diversity of these populations that we're dealing with. Skin, all sorts of bacteria. Some of them you might recognize the names. Staphylococcus, for example. There are several species of Staphylococcus which just occur naturally on our skin. In the eyes, we do have a resident bacterial population, but it's very much kept under control by lysozyme, the substance in tears. So the populations are actually low, but also quite diverse. The nose and the upper respiratory tract, nose, sinuses, have a very rich microbiota, but the lower respiratory tract does not. The lower respiratory tract is kept largely clear of bacteria because of that escalator of mucus that moves from the lower parts of the respiratory tract up the throat, continually sweeping out bacteria which make their way any lower than about the bronchi. So mouth has a huge population of bacteria. Large intestine and the small intestine, these have literally thousands of genera of bacteria present. You don't need to remember any of those names, just to be aware of the huge diversity of our microbiota. Now, we keep saying that this microbiota is protective of us, and we need to understand why. The first thing to realize is that bacteria are in competition with one another the whole time. They are competing for space, and they're competing for nutrients in particular. And they actually can exclude rivals for those things by various stratagems. But in general, it is simply the size of the population that acts being the major excluding factor. Bacteria simply can't find the space to grow. Bacteria, they need to be attached. They need to be attached in a suitable environment, and they need to be attached somewhere where they can acquire nutrients. A rich population of healthy bacteria on our skin, in our gut, in our mouths, and elsewhere actually exclude pathogens. They simply don't have the space to grow, and they don't have, even if they were to establish, they don't have the resources to develop their population. So what they're doing, they're competing for nutrients. The bacteria of our microbiota may, in fact, produce substances which are harmful, antibiotics, which limit competitive bacteria. They change the environment. They make it unsuitable by altering pH, by altering the amount of oxygen, etc., etc. It has to be said, though, that some of our microbiota may occasionally become opportunistic pathogens. This one here is really famous. This is Staph aureus, Staphylococcus aureus. Staphylococcus aureus is a normal part of our microbiota, but it occurs in very low numbers. You can isolate it from anybody's skin, but in low numbers. It's just that under certain special circumstances, Staph aureus can become a pathogen. It enters a pathogenic kind of phase. For example, if you have a wound that becomes infected, Staph aureus is a very likely bacterium to infect a wound in the skin. Staph aureus is also the bacterium that most commonly causes pimples to become boils, to become large infected areas. So it can, under certain circumstances here, Staph aureus can become pathogenic. It has to be borne in mind that some of these bacteria, if the balance is affected, may in fact become opportunistic pathogens. Okay, let's just for a minute just talk about these kind of interactions that we have with other microorganisms. There are three terms that we need to get clear before we start to talk, for example, about disease. The first term to understand is commensalism. Now, all of these, there's a word that covers all of this, and that's the word symbiosis. Symbiosis means living together. There are different ways of living together. Commensal organisms, this does apply to a lot of our microbiota. These are organisms which benefit from their association with us. They do us no harm whatsoever. In fact, they don't affect us at all. However, remember that that applies to particular organisms, not to the whole microbiota. The whole microbiota confers tremendous benefit to us. Individual members of that microbiota, we may not be able to see a particular benefit to us of their being there. They benefit from us, but we are unaffected by their presence. That is the typical commensalism. A good example would be the staphylococci that we have on our skin. In mutualism, both parties benefit. This is the case for much of our gut microflora. We now understand that we are, although we cannot always see the benefits that we get from a particular bacterium, it appears that we derive tremendous benefit from these diverse bacteria in the gut, for example. An example would be E. coli bacteria. And E. coli bacteria, very, very common. You've probably heard of them already. They're a common member of the gut microflora. And not only do we provide them with a warm safe habitats to grow. We provide them with lots of nutrients. They, in turn, give us various products, and in particular, they contribute to our vitamin budget. And vitamins, B vitamins and vitamin K are all derived from organisms like E. coli in the gut. So we can actually pinpoint specific products that they produce that we benefit from. The rest of our discussion, actually for most of the rest of the semester, is going to be discussing the role of parasites. And this is a symbiosis where one organism benefits, but it's at the expense of the other. The other organism does not benefit. In fact, its homeostasis is adversely disturbed by the presence of the parasite. And this definition applies to all pathogens. So, for example, we regard, even though they are not alive, we regard viruses as being parasitic, because the benefit to them is we end up reproducing them, and they detract from our homeostasis considerably. Pathogenic bacteria, it's the same story. They are using us as a host. They are deriving benefits of habitat and nutrients from us. And in turn, they are actually damaging us. They are disturbing our homeostasis adversely by their presence. So that is the definition both of parasitism. And then a pathogen, really any parasite occupying us is occupying a pathogenic role. But we really think about pathogen as being a parasite that produces actual disease, that produces a course of disease. I make that distinction because we may in fact have parasites that barely make any difference to us. Think, for example, intestinal worms. We tolerate intestinal worms very, very well. They make very little difference to us. It is true they are using nutrients that we could otherwise use. So they definitely are parasites, but they're not necessarily going to cause a disease process to begin. For most of the viruses and the bacteria that we're going to be talking about in the rest of this semester, that is not true. The parasitism of these organisms actually causes a disease process to occur with very distinct symptoms that we can follow. These, by the way, are influenza viruses being grown in culture, but you can see them budding out of this cell here that they've infested. Okay, so I just want to remind you, please, about, we did discuss this before, but it bears repeating. And that is that microbiologists actually use a very strict definition to decide whether or not an organism, or anything, a virus is not an organism, but in any case, whether an organism is actually causing a specific disease. And these standards were established by Koch, who, the German microbiologist in the 19th century. He originally devised this to describe the disease caused by an organism called Bacillus anthracus, and that is the disease of anthrax. He wanted to prove, it had not yet been proven, he wanted to prove that bacteria could cause disease and that they could cause a specific disease. And he had been working on anthrax, which at that time was a tremendous problem, especially in cattle, in farming. And so this was the organism he worked on. So here he established these, which are known as Koch's postulates. And if you're going to study microbiology, this is very, very important that you understand these. First of all, he actually started this work in the field, working with animals which had died because of anthrax. And he started wondering about anthrax because he extracted bacteria from dead animals. But now he's got a dead animal, he's got bacteria that he's extracted from. Can he show that those bacteria actually caused the disease that the animal died of? What he did was he established culture mechanisms whereby he was able to grow the bacillus in culture. That was important because it meant that he got a pure culture. He only had one kind of bacterium growing. He grew them on plates. He grew them as colonies. So each colony is billions of cells, which are genetically identical. They are a clone. So he knows he's dealing with a pure culture. He then took those bacteria and he inoculated healthy animals with the bacteria. He observed the animals very carefully and followed their symptoms. And he saw that they reproduced the symptoms of animals that had died in the field. So these animals behaved the same way, for example, that the cattle did. They had the same course of disease and they then died. He was then able, when he went and looked carefully, he was then able to extract bacteria from these animals and purify them. And he was able to show that these bacteria were exactly the same as these bacteria. So the bacteria he cultured here were the identical bacteria to these. So these are Koch's postulates, that you must be able to extract the bacterium and purify it. When injected into an experimental animal, the disease must be reproduced in the animal. You must be able to extract bacteria from the experimental animal that has suffered the disease, and you must be able to purify it. And when you purify that bacterium, it must be the same as the bacterium that you originally identified. And if you satisfy those postulates of Koch, you can say this bacterium causes this disease. It seems simple enough. And it actually is still accepted as the standard for establishing the etiology of particular diseases. But there are some problems. This is we often just instinctively use Koch's postulates when you're investigating a new disease. But there are a few problems with using Koch's postulates. The first is, if we go back, it is possible, and it really does happen, that the same bacterium can cause a different disease. So in using anthrax, he extracted originally from cattle and then purified and used experimental animals and mice. And so, well, that's fine, but the same pathogen can actually sometimes cause different diseases. And then you get hung up on this step here, because you're not reproducing the disease that you originally saw. So that's the first thing. The second thing is that we have to have a reliable animal model in order to do this. So, for example, if you will still hear people say that it has never been proved that human immunodeficiency virus causes the disease of AIDS. And you'll hear people say, well, it's never been proven because Koch's postulates have not been satisfied. You can't do that for a human disease because you can't experimentally give somebody the disease. That is unethical. You can't do that. Okay, so there in human diseases, in purely human diseases, which HIV is, we have no good animal model for HIV. In purely human diseases, we do run into difficulties. The third difficulty with Koch's postulates lies at this stage here, and it's a very important one, and that is not all bacteria, for example, are culturable. They may well be alive here. We might be able to extract bacteria from a dead animal and inject it into another animal, get that animal infected, but we can't actually pure culture and purify the bacterium to study it and to establish these two steps. So sometimes Koch's postulates, we have to just do the best we can to establish the chain of evidence and then make our assumptions after that. I would mention, as far as the story with HIV goes, that is this here, although we didn't do a deliberate experiment to show that HIV followed Koch's postulates, unfortunately, an inadvertent experiment was done. And that is if you trace people, for example, who use injectable drugs, who used contaminated needles, they in fact passed the virus to one another as if that was through an experimental chain. And that can be traced because we can look at the genetic makeup of the viruses that they are infected by and show that there is the identical strain. So we have an original person who's infected. They contaminate needles. They infect other people, and they suffer from exactly the same disease. And we then go look at their virus, and we can show that it's the identical strain that the original person had. So this unfortunate inadvertent experiment now satisfies Koch's postulates, unfortunately. Okay, so I warned you that we do have to learn proper definitions for some of these things, some of these words, because some of the words are so familiar to us. We use them in common parlance, but they have a very particular scientific meaning, especially when we're talking about disease course. And the first one is this word symptom. A symptom is a specific change in body function that results from disease, from a disease process. It has to be a specific function. And so just for example, I don't feel well is not a symptom. I have a lateral headache on this side of my head, which has persisted for 6 hours. That can be a symptom. A symptom is something specific, and it is tied to the disease course. The signs are different. They are the signs of illness, of disease, are changes. They can be confused with symptoms. For example, change in temperature is a sign. It's a sign actually of immune activation usually, but it may also be a sign of toxin production by an organism like a bacterium. But that is a sign. It is an overall sign that the body is responding to a disease process. A syndrome is a collection of signs and symptoms that predictably are associated with a particular disease, not necessarily exactly the same in all cases. So, for example, the disease caused by HIV is acquired immunodeficiency syndrome. It is a syndrome because all sorts of signs and symptoms arise due to the collapse of the immune system. They may not be exactly the same in two different patients, but they have similarities. And if we look at the whole population of patients, we see them regularly reproduced together. And so they form a syndrome. Okay, so we are largely going to be talking about communicable disease. In the case of both viruses and bacteria, most of the disease that is caused is communicable. That is, it can spread from one host to another by whatever mechanism. So it is communicable by one means or another. A contagious disease is a communicable disease, but it is spread directly from host to host. So, and the contagiousness can vary tremendously. Some are not very contagious. Others are extremely contagious. So a communicable disease is any disease that can spread from host to host. That's how disease maintains itself. But that communication can be direct, person to person, or it can be through some other agency. A contagious disease implies a person-to-person contact, with the disease being spread directly from one host to the other. A non-communicable disease is a disease which is not spread from one host to another. Many parasitic infections, for example. I've given you some examples which we will hear about later. For example, many diseases are communicable, obviously, but I give you this one, tuberculosis, as an example, because tuberculosis is definitely a communicable disease, but it is not very contagious. It's not that it's not contagious at all. It is contagious, but it is not very contagious. On the other hand, a disease like measles is highly contagious, very, very, very highly contagious, very, very easily spread from person to person, which is one of its great dangers, as we'll learn. So on with our terminologies, but this is one which I really want to hit on. This is the word incidence. Incidence has a very particular meaning in disease process and in epidemiology. An incidence is a rate. Now, commonly, unfortunately, you will hear people mix this word up, incidence, with incidents, I-N-C-I-D-E-N-T-S. They are not the same thing. An incident is something that occurs. If several things occur, there are incidents. But if we measure how often they occur in a specific time, we can say that that is an incidence. An incidence is a rate. It's the number of times that something particular occurs in a specified time. In epidemiology, at the moment, our interest very often is on 7 or on 14 days. We look at 7 and 14-day incidents of COVID-19 hospitalization, for example, as a measure of disease. So that is an incidence, it is a rate. Prevalence is how many people develop the disease. And so we can say the prevalence of this of COVID-19 in the American population is, I forget what it is now. It's like 2 million or whatever, 2 million persons in the United States. That's a prevalence of the disease. It is not a rate. It's not an incidence. It just tells you the number. And you can specify the prevalence. You can sometimes say the prevalence has increased over time, but that's still not giving you an incidence. It's not giving you per time. Okay, so we'll see disease itself referred to by various monikers. For example, a sporadic disease is one which only occurs very occasionally. It usually has a very specific localized source, and it is usually the number of cases is usually self-limiting. So it occurs suddenly, usually. It may be quite spectacular. A lot of people may get it in a circumscribed area or whatever, and then it dies out. And then it occurs again unpredictably somewhere else or whatever. A typical example would be this disease, Legionnaire's disease. Legionnaire's disease is caused by a bacterium called Legionnaire, which develops in water. And when that water is not vaporized, but when it's turned into a mist and sprayed out, for example, from air conditioner units, this sort of thing, and sprayed into the air, if people breathe it in, they acquire the bacterium and they can suffer from this disease, Legionnaire's disease. So Legionnaire's disease is often limited to a particular air conditioning unit, a particular building, a particular set of buildings. It occurs suddenly, and then after however long, it dies out, does not spread, it's not contagious. So it's purely sporadic. Endemic disease is a disease which is constantly present in a particular area, often at a very low rate. Often it is maintained somehow, either in the human population or another population, and then and it flares up from time to time. And what is very important about endemic disease, for example, cholera is endemic to the country of Bangladesh. It occurs in its waterways, for example, and it is just part of the natural ecology there. And it is often present at a low rate. You can usually find some cases here, some cases there, and then periodically it will bloom and become epidemic. What is very important about that is that it may spread out from the endemic area and begin to move into areas where it is not endemic. So an epidemic disease is one which is acquired. Let me just say this. In an endemic situation, for example, you often find a baseline level of infections present. Epidemic is where the number of cases suddenly increases from a baseline in a specific area. So, and an epidemic is usually described by geographic area, by country, maybe by town, maybe even by school or something like that. You'd say there's an epidemic when numbers suddenly increase above what you would normally expect. So, for example, we would say that measles epidemics are becoming increasingly common in towns where, for example, vaccination is unpopular. This is a specific thing. Anti-vaccination organizations tend to operate in specific areas where they are successful. We can usually find epidemics of the diseases associated that have not been vaccinated for. So this is in a specific area, for a specific time, and the number of cases suddenly increases above what may have been a very low background rate. That is an epidemic. Pandemic, which is what we are witnessing at the moment, is when these epidemics spread from geographic area to geographic area, and they do so on a worldwide basis. And that is what we're watching. The pandemic spread of COVID-19 began as an epidemic in China. It was then spread to other areas by various means, by travel, etc. And in those areas, it became epidemic, but soon it began to spread out from those epidemic areas to the worldwide pandemic that we see now. So we can see a chain of events. We might look at it like this, that in fact, the COVID-19 is endemic to that area of China. It is endemic there because it's actually resident in animals. It's then spread to humans in China and became epidemic. The epidemic in China then seeded other epidemics elsewhere, and before long, there was a runaway epidemic worldwide, which is the pandemic. So let's talk a little bit about the course of disease, because this is going to become increasingly important as we talk about specific diseases. An acute disease is one where symptoms develop rapidly, and it is usually characterized by having a circumscribed length of time, and the person either recovers or dies, but the symptoms, the signs and symptoms abate as the disease is overcome in an acute situation. In a chronic situation, symptoms are present over a very long time. It is true that chronic disease may arise as a result of acute disease. So, for example, with COVID-19, we're discovering that people suffer an acute illness and appear to recover from the acute illness, but then suffer from chronic symptoms over an extended period of time, and so therefore end up suffering a chronic disease. Some diseases are simply chronic. They seldom have an acute phase. Subacute disease, this is in between the two, and the problem with this is that people often are suffering such mild symptoms that they remain completely functional, and they may actually be agents of spread of the disease. This is very similar to them having latent disease. And latent disease, the pathogen is present, but it is not causing symptoms. It does not cause them to feel that anything is wrong, but it may then later on activate and produce symptoms, but it is present, and people either in subacute or in latent disease run the risk of being carriers of disease, of actually spreading the disease without knowing that they're doing it. Now, I don't want to have a word about herd immunity, but I think I'm going to leave it to a little bit later, because I have some slides to show you on herd immunity. That we want to have a specific discussion about herd immunity. I'll simply say this for the moment. Herd immunity is a term which is particularly applied to situations where we apply vaccination very widely in a population, and we make enough people in the population immune to disease that the pathogen itself has no route to somebody who can be infected. The pathogen has to be transmitted to somebody who becomes infected. And if the pathogen cannot make its way through a population because everybody's immune to it, it protects people who in the population who are not immune. The pathogen simply can't reach them. That is what herd immunity is, but I'll have more to say about it in a while. Okay, so at the very beginning of our microbiology class, I told you about this early scientist called John Snow. And John Snow was a scientist who worked in London. And he was the very first person to apply epidemiological principles to the study of disease. The disease he was interested in was cholera. Cholera is a waterborne disease. It causes a very dangerous and very violent diarrhea. Nowadays, it's much less dangerous than it used to be because we can treat with intravenous fluids and everything, and people tend to recover. But in the old days, it was regarded as a plague disease, and its cause was completely unknown. But John Snow decided to work on cholera outbreaks in London, and he applied the earliest applications of epidemiological principle, because what he did was he went and found out who was ill. He accumulated data about when they had fallen ill, how many people in their household had fallen ill, and where the houses were where people were falling ill. There was an outbreak in a poor area in the east of London, and many people died. He mapped out the distribution of houses where people were infected and had died, and he realized that they were all using one common water source, which was a pump in a square. And the houses described a neat circle geographically around that pump. And he reasoned that what was happening was, in those days, they didn't have much water. There was not a lot of waterborne sewage, especially in the poor areas of London. Instead, people had backyard privies. They dug a hole in the ground as deep as they could go, and that was the lavatory where people went and defecated. But the problem was that these privies all communicated with the underground water source. And if you get somebody who has cholera and they contaminate the water source, the cholera bacterium spreads through the water source and gets picked up at the pump and now gets distributed to everybody who uses that pump. And John Snow realized that that was what was happening. And he begged the authorities of London to please close this water pump and bring water in from somewhere healthy. And they refused. They said this was nonsense. The germ theory had not yet been proven. They thought it was a ludicrous idea that the water could be causing the illness. So John Snow very courageously snuck in there in the middle of the night, and he stole the handle of the pump, so that in fact people stopped using the water and they had to go elsewhere to collect water, and they mostly collected from healthy water sources, and the epidemic died down. Our very first documented, he wrote all this down and wrote it up, our first documented true epidemiological study of a particular disease, describing its etiology and describing its provenance, how this disease actually came to be imparted to all of these households at the same time. So what an epidemiologist does, they determine the etiology of the disease. They look for all of the important factors that concern that spread, what is amplifying its spread. They develop methods for controlling the disease, largely by looking at this, largely by looking at how the disease is being spread. John Snow, people are collecting from the water pump, contaminated water from one particular water pump, close the pump down. and you close off that source of infection. And very, very importantly, epidemiologists assemble data and they assemble graphs and they model, they do mathematical modeling of disease spread in order to explain what is happening and to make these recommendations to control the disease. Exactly what we are seeing from the responsible members of the CDC and the other government agencies here and elsewhere in their attempts to explain the spread of COVID-19 and explain to people what we ought to be doing to limit, to control the disease. Realizing that we're dealing in COVID-19 with an extremely dangerous disease, not because it has a very high mortality rate. It doesn't. It has a higher mortality rate than influenza. Not for that reason, but because it is so contagious. It is so very, very contagious. That is its huge danger. It spreads very rapidly to an enormous number of people. And that means it spreads to vulnerable people very easily. And that is its huge danger. The major thing to know about epidemiology is a data-driven science. It is a scientific endeavor. It is not a matter of opinion. It is not a matter of politics. When an epidemiologist says wear a mask, it is for a specific reason. And it is because they understand the etiology of the disease. The disease is droplet spread. COVID-19 is droplet spread. The epidemiologists have been able to show that very clearly. So they can make the recommendations: wear a mask, socially distance, and wash your hands. We're all of us now in the middle of an epidemiological crisis. If you want to think about it more positively, we are subjects of an epidemiological experiment. And we hope that the outcome of the experiment is successful. Here are some, just to illustrate for you, the sorts of graphs and things that epidemiologists would produce in order to try to understand disease. This is the disease of HIV/AIDS. These are actually the number of cases that were recorded, and this is a positivity. So first of all, look just first of all at this initial part here, there. Look here and you'll see that this block here are the first 250,000 cases recorded in the United States. So these are all people who have turned up HIV positive on blood tests. The disease enters somewhere, the population somewhere around here. It moves through particularly vulnerable populations. But if you follow its growth, you'll see, doesn't that look suspiciously like a growth curve that we saw earlier when we looked at bacterial growth, for example? There are reasons for that. There are reasons why this growth curve looks like that, and that is that the virus in this case is spreading through a vulnerable population, and it is initially going through an exponential growth phase. A terrifying experience when a new disease suddenly appears in a population, because the disease moves through the vulnerable population with great rapidity. So have a look here and you'll realize this is probably 1980 to 1992, 12 years for us to accumulate our first 250,000 cases. But the next 250,000 cases were accumulated in only 4 years. This I can promise you, because I was there. This was a terrifying time. It was a terrifying time, especially because we didn't understand until somewhere around here what was actually causing the disease. But the epidemiologists were able to track it because it had its specific signs and its specific symptoms, and was very quickly recognized as a syndrome. And so that was what alerted everybody to the fact that the disease was there at all. Down here, there were very few people with the disease, but they had very particular markers. And those markers were the collapse of the immune system in young people, in young, healthy men, for example, something completely unknown. And from these very early few numbers, within 12 years, we had accumulated 250,000. The disease by this time was rampant. But at this stage, there was little known about how to deal with it. There was no therapeutics for it. And the death rate was very high, but public awareness became enormous by the 1990s throughout the vulnerable populations. There was enormous epidemiological effort to get people to reduce their risk. And this is reflected. I showed you here 250,000 cases in only 4 years. Look at the next 250,000, 1, 2, 3, 4, 5. It's beginning to increase. 1, 2, 3, 4, 5, 6, 7. We're beginning to see an expanding length of time to accumulate the cases. This is a reflection of epidemiological effort to get people to reduce risk. I told you this is actually these are actually positivities. If we were to look at the death rates, we would have seen that the death rate would follow this very closely up until the end of the 1990s, when the death rate would come right down, when therapeutics were introduced. Around about here, successful therapeutics were introduced. Very, very successful treatment is now available. The death rate from HIV has decreased, but I do want to, before we congratulate ourselves, I just want to show you, look at this 250,000, these 250,000 cases. The time is decreasing. The time to accumulate 250,000 cases is decreasing. The infectivity rate is increasing. The incidence is increasing again. Why? Who talks about HIV these days? Who worries about HIV? Who in our sexually active young population even thinks about HIV these days? The way everybody did that back here when we were all terrified. Now it's a thing, almost like it's a thing of the past. This decreasing time should tell you, and the epidemiology tells you, that it's not a thing of the past. It's something that has to be paid attention to. And as far as HIV is concerned, we still need to pay attention to the strictures that we applied here, safer sex, etc., etc., etc. The virus is still there. Even if we have good therapeutics now, it's not something that one wants to get unnecessarily. Okay, let's have a look at some other epidemiological graphs. And these, of course, are for COVID-19. So this is our epidemiological situation right now. And sorry, this is not. I'm going to talk about COVID-19. These are just graphs to show you the different kinds of epidemiological statistics that we use. This is for Lyme disease. Lyme disease is a tick-borne disease, and it did definitely show an increase in numbers, a sudden increase in numbers in the early parts, 2007, 2008, around about there. There was a sudden increase in numbers. Exactly why, nobody is absolutely clear. But here you can see, here is one statistic frequently reported in epidemiology, simply the number of reported cases, usually for a specified country or a specified geographic area. This is the United States, and you can see there is a background level around 15,000, but there is a slow and then a rapid increase in the number of cases. Then decrease, and it sort of bounces around. This is not clearly explained. There are various things that have been suggested. One of them is people, suburbs moving into wooded areas, houses being built in remoter wooded areas, and people being exposed to the ticks more. But the etiology is not absolutely clear for Lyme disease. We know exactly how it is caused. It's caused by a bacterium called Borrelia burgdorferi, which is injected into you by a tick bite. And in some people, it's just a mild transient illness. In other people, it forms a chronic condition and often associated with arthritis. So this is a simple numbers by time, numbers by year or whatever. Here's another, this is a much more common way for epidemiologists to report numbers. And this, for example, is the number of cases per 100,000 people. You may also see other figures. Always pay careful attention to the y-axis, because sometimes it is reported per 10,000, sometimes 1,000, etc., etc. Make sure that you understand what the y-axis units are. This is the reported number of tuberculosis cases, 1945 to 2016. And it's really interesting because have a look here and you'll see that, that tuberculosis in the late 1940s was very, very high. If we went back here, it would be around, it'd be bouncing around like this. So this is the end, actually, of a very long time when tuberculosis was absolutely endemic in the United States. Not epidemic, but endemic. It existed at a constant rate, quite high in the population, of about around about 100 per 100,000. And tuberculosis, it's caused by a bacterium, Mycobacterium tuberculosis. There's very little that could be done for people with tuberculosis here. Mostly they try to have lifestyle changes. They sent them off to high altitudes to live. They sometimes would collapse a diseased lung to isolate it. But the fatality rate was actually quite high. So this is not deaths, this is reported cases. So have a look here, about 1949, something spectacular happens. And that is we get the first antibiotics, which will hit tuberculosis. And immediately the numbers begin to drop. In addition to this, there was a tremendous amount of effort, epidemiological effort, put into educating people about tuberculosis and trying to limit its spread. It often spread within families. It's not very contagious, so you need extended exposure, and very often it would spread in families. So a lot of effort went into public awareness about tuberculosis, and then this spectacular impact of therapeutics reduced the number of cases. in addition, once we started to treat tuberculosis, we reduced the number of people who were able to spread the disease. So that is another aspect to this decline. And here we are. This is where we are today. This is our current pandemic. This is looking at our actual case numbers. And this is for New York. And this is just case numbers reported straight. And we can, this is this year, and you can see the typical epidemic spread. That absolutely, of course, it did not exist in the population at all until the beginning of this year, entered the population, and very rapidly undergoes exponential growth in the population as it rapidly spreads into a vulnerable population who at this stage, we were all naive. And that, I mean, immunologically naive. We had no resistance to it, and yet we were doing nothing really to protect ourselves because we simply didn't know it was there. It wasn't clear what was happening, and the numbers spread very, very rapidly. There is a problem with this kind of data, these kind of data, and that is this. You can see all the spikiness here. This is largely due to reporting error, to different people reporting cases at different times, different hospitals reporting. Some maybe save up all their data for 3 days and then send it in. Others report every day, and the data overall is not incorrect. It's just that it accumulates all this noise, which can make it difficult to interpret. We had in New York had a very, very bad spring where there were an enormous number of cases, thanks to the fact that in our state, people have tended to listen to the science. People in New York have been exceptionally good about wearing masks and socially distancing. Everything closed, shut down to limit social contact. So they limited the spread of the disease, and we saw a spectacular decrease in the number, despite the fact that the disease is rampant elsewhere. This is the impact of good epidemiology practice on a responsive population. A very big decrease. However, we went through summer, we evaded a second wave in summer, but we are in grave danger now. We are seeing an increase. People are tired. They're tired of wearing masks. They're tired of not going to restaurants. They're tired of not going to the bars. They're tired of not seeing their friends. And in addition, we have high incidence in other parts of the country, which act to seed our population. And many of people in our population are making themselves vulnerable once again. And this is a very worrying trend. So